part was from the lab, of course, and then we'll add in a little bit of the hormones. So we'll kind of go just super quick. Of course, we're just going to start off with male, which is a headphone. All right. You've got, of course, ducts and glands when we look at this, and then we have the testis with the scrotum here, of course. So the testis, of course, is where your sperm is produced, and then once that sperm is produced, it has to go through a maturing process. So it takes 30 to 60 days for the sperm to actually mature as they go through this. And they'll be produced inside the testes in the seminiferous tubules, and we'll talk about that process here in just a few minutes. And then they make their way into the actual duct here. And so the very first duct they're going to enter into is the epididymis, which is a coiled kind of comma or C-shaped duct that lies on the posterior and anterior portion of the testis. And then from there they enter into what's the older school term is vas deferens, or the duct deferens, which travels up into the pelvic cavity, courses over the top of the urinary bladder, and then is going to merge over here and to form the ejaculatory duct. Well, this part right here that's enlarged is referred to as the ampulla of the vas deferens or ductus deferens. So that's that duct portion there. Then we have, so this would be the sperm are produced here, they're going to travel through here, and then we have fluids that need to be added to them to produce the semen. And so we're going to have the prostate gland here, which produces a prostatic fluid. It's about 60% of the semen comes from the prostate gland, and you can see that here is also the seminal vesicle, and the seminal vesicle also produces another gland. And so between the two of these, we're adding enzymes, we're adding nutrients, we're adding sugar, we're adding the majority of the volume here would be coming from those two glands, and they all basically merge together here. And so you could call this, of course, the prostatic urethra at this point as it goes through the prostate gland. It goes through a little area where it's actually not in anything, so a lot of times they'll call it a intermediate urethra or a membranous urethra, but there's a little area there. And then once it courses right through this general area, we have another gland, our third gland, the vulvourethral gland, which produces a neutralizing mucus to basically help clear the way, to neutralize any acid from urine that has been in the urethra, and it kind of just clears that out so that the semen as it comes through doesn't have to deal with the acidity there. And then, of course, you have then the urethra, the spongy urethra, with the penis, etc. We had talked about in lab two muscles that were associated with the scrotum, the dartus and the cremaster. The dartus crinkles the scrotum, and the cremaster pulls the scrotum up close to the body. I may have said that backwards. I never can remember. Sometimes I do, and I don't know why. But anyway. So those are those two muscles there. This is looking, just zooming in on the testis and the epididymis, so you can kind of see that a little bit better. And you can see that the testis is divided into a lot of lobules here, and each little lobule then has a tubular system in there called the seminiferous tubule. Now, we looked at this under cross-section, and so it looks very similar to this right here. This is the drawing, here's the actual thing. So you actually have the outline, line of the actual tubule, and then this is where our sperm are actually produced. So this is our site for meiosis, which is a special reduction division that cuts the chromosome number in half. And so all of the sperm that are, of course, produced here have 1/2 the normal chromosome number. So instead of having 46, they have 23. So that when the egg and the sperm join together, we restore back to our 46 normal chromosome number. You would also have along here cells that are going to be involved in producing some of our hormones. And you're going to have support cells here as well, and we create a blood testis barrier here as well to protect those developing sperm from the male's immune system, since they would not be identical, because not only have we cut the chromosome number in half, but we've shuffled the chromosomes up as well. So there's been a mixing of the chromosomes. So those chromosomes are not like the original adult. So they would definitely be perceived as different. So you can see, of course, the developing sperm there. whoops, sorry, let's see. This just goes over the basic structure. We've kind of already pointed this out. We're not doing all the various parts of the penis, the root, the body, the glands. We're not going to do that. We're just at the penis. And moving on. Here is, of course, just a diagram of the spermatogenesis itself. So production of sperm, spermatogenesis. So here, and it shows you the original cell that was to begin it all is the spermatogonium. Here begins with 46 chromosomes, and then we're going to undergo, begin our process here, and so you can see meiosis 1, meiosis 2, and so now we're finally getting down to our developing spermatids here, which they're incomplete. You can see they're not quite fully mature versus a little bit more mature spermatid down here. Once they're activated and can actively swim, you can just call them sperm at that point. We tend to use the term spermatid to imply that they're still developing kind of step with it. And so these are produced all the time. Sustenticular cells are going to be one we'll refer to here in a few minutes, and interstitial cells. These are going to be cells that help play a role in the release of hormones and things like that. So these are going to be cells that are targeted by our hormones like follicle-stimulating hormone and luteinizing hormone and things like that. So we should have a picture. This is just showing you the maturing of the sperm. So it is kind of good to know. The sperm is really just a delivery package for genetic information, just like a little smart missile there. So you can see it doesn't have a whole lot of organelles associated with it. A lot of the excess cytoplasm is pulled off as this develops here. And so you can see here like the Golgi apparatus and various other organelles are kind of pulled off. So we're really just down to the actual nucleus with a small cap over the top to help protect it, the acrosome. Then you've got the little mid piece here, and then you have the flagella or the tail. So really just a delivery system for the genetic information, not any support material or anything like that. And that's why the eggs are so much larger, is the egg contributes the cytoplasm, and all those organelles as well would come from the female's contribution, because the male's contributing just the DNA there. Okay. All right. So let's look at the hormones then. So when we begin with this, and you can see there's several step process here, like a three tier process here. For it to all begin, it begins, of course, up with the hypothalamus, and the hypothalamus releases a gonadotrophic releasing hormone, which just means it's going to direct the pituitary to release hormones associated with our development of reproductive organs here. So hypothalamus triggers it off at the very beginning, stimulates the anterior pituitary, and so it releases two hormones that we've already learned, the follicle-stimulating hormone and luteinizing hormone. So both of them will go and target the testes. But here specifically, if you actually looked at it, luteinizing hormone is going to actually stimulate the production of testosterone. And this testosterone is coming from what they call the interstitial cells. So these are the ones around the outside of the seminiferous tubule. So those are really the ones responsible for the testosterone. Now what we had said when we did endocrine, we said luteinizing does the hormones and follicle does spermatogenesis. And it kind of does, but it's a little bit more complicated. Our sustenticular cells here, the ones inside the seminiferous tubule, then these guys are going to be stimulated by follicle-stimulating hormone, and they actually secrete a product called ABP, which is androgen binding protein, which binds to the testosterone that's been released. And then that modified testosterone will then stimulate, kind of comes back around, stimulates the actual spermatogenesis and the developing of the male sex characteristics. So let's do this one more time. Okay? So we know hypothalamus stimulates the and you don't have to know gonadotrophic releasing hormone. We just say hypothalamus tells the pituitary release follicle-stimulating hormone and luteinizing hormone. Luteinizing hormone stimulates the production of testosterone from the cells around the seminiferous tubules, not inside it, the interstitial cells. Follicle-stimulating hormone then
stimulates the cells inside, the sustenticular cells, to release a androgen binding protein, which binds to that testosterone right here. So here you got both things being produced. They bind together, and now our newly modified testosterone stimulates our spermatogenesis and our sex characteristics for the male. Pretty straightforward, actually, when you compare it to the females, which is a nightmare. You know, when they say, oh dear God, this hormonal well, because it's all over the place. So pretty straightforward as far as dividing them up. You can still simplistically say luteinizing produces my hormone testosterone, and follicle is responsible for spermatogenesis, but we just added just a little bit more in there to that. Okay? Yeah. All right. Now, okay. So I hate to minimize, but boom, that's it on the male. So just because we've got several things we want to get through to. So with a female, basic structure, of course, of the uterus, we have the uterine tubes or fallopian tubes leading to the little finger-like or fringed-like fimbriae, which basically almost touch, not quite touch the egg-like ovary, which is a nice oval structure, which is then suspended by that ovarian ligament there. Sits on top of the bladder. When you look at the uterus, you do have the three layers, the endometrium, the myometrium, and the perimetrium. Endo being the lining, and myos the muscle, and peri is the outer part there. You have several ligaments. The ovarian ligament suspends the ovaries. Then you have, I think in lab we're just doing the broad ligament, which then is kind of like it exactly says, it's a broad sheet that helps to suspend and hold the uterus in place, so that it doesn't basically collapse and fall out and cause prolapsed uteruses, which we sometimes see. The uterus itself, big difference between human uteruses and animal uteruses is in animals they tend to be horn or Y-shaped, so because they have multiple births, so they would have, they'd come up this way, they'd come up that way, and then of course in a human they're not shaped that way, so they're not designed to handle multiple fetuses. So twins, yes, but you end up with a lot more risk with triplets, quadruplets. Those things are all much rarer, and part of it has to do with too is that the uterus is not designed to hold multiple fetuses in here, because we need a different design if we're going to always have a litter of children, which we don't, thank God. Anyway, so same picture, we just kind of looked at that already. Do y'all know what a prolapsed uterus is? I don't know why I'm bringing that up, but it's happened. This has come up in prolapse. It's like everything's from inside is falling out. So it prolapses, it comes out. In animals, it comes completely out. And in a human, it usually we don't normally see it coming completely out, but they might go in and you'll actually see a weakening of the walls here, and you can definitely see that it's just dropped from the position and it's coming out, but it doesn't normally completely evert like you would see with an animal, but it's not a pleasant thing. Put that stuff back up. Anyway, all right. Sorry, I just had a really long conversation with somebody about prolapsed uterus, and it was something. What was it? Somebody's cow. Anyway, all right. So this is oogenesis. So oogenesis is the production of an actual oocyte or an egg. Okay. The big difference here is that males, spermatogenesis is happening 24/7 once we've reached puberty and continue on. They're producing sperm for continually, in huge amounts of sperm, really. With a female, these are pretty much done it's kind of determined, this first step, before birth, as you can already see. This has already happened before the person's born. So we have essentially a finite number of eggs, and then of those, only a few of those are going to actually get selected for, and a lot of the other ones just get reabsorbed by the body. So we have a much smaller one, and we have a lot of arresting of development, as it were. And to go through this total process, this is the fertilized process here. So a lot of it doesn't continue until you get right down in here. So we kind of start the process and then we stop it. And we start the process and we kind of freeze it. So there's a lot of pausing in this process, which means that the stage is really set for more mistakes to occur because it's not one continual process. You have a lot of pauses here. So meiosis, again, the oogonium is 46 chromosomes. And so we begin the process of meiosis, or dividing. We begin that process way up in here, and then we pretty much stop it here in one of the very first phases. And so that's where it stays, like this. So you can kind of see here during childhood to puberty. So puberty-wise, you can see we begin to see some development here of some of the follicles. They go from primordial to primary, secondary here. So at this point, this person would be, obviously, menses would have begun here if you're seeing this happening here. But this would be our normal progression here. This would be a follicle that's ready to basically ovulate or release our egg here. Here you have ovulation. Here's your little secondary oocyte at this point, and still some of the last bits aren't going to happen until right in here, because like right here it says arrested in metaphase 2. So we're still not through with meiosis even when we get to this point here. And it doesn't happen until we get on down a little bit. Now, the other thing that, I don't know if I have a count that shows this. Let me see. No, I don't, but that's okay. So think of it this way too. With a male, in males, you have your original that was 2N. So every one you're going to end up with 4 sperm, right? So 4 functional spermatotids from one division here. With the female, you start here, you bring it down, you end up, oh, how do I even describe this? You end up with these little guys. These are called polar bodies. And we get one functional egg. So for ours, we don't get the same numbers. So because we get a lopsided cytoplasmic division. Lopsided cytoplasmic division. So that's what you're kind of seeing. They're showing you a little one of them right in here. It says first polar body, and it degenerates, which means it just degrades down the body, just absorbs that. And there'll be, here's another polar body here, it gets absorbed as well. So what we end up doing is we're taking all the cytoplasm, the organelles, and we're shifting them to one primary cell with each one of those divisions, so that that one ovum is large and it has all the different organelles that you can possibly have, lots of cytoplasm, everything in there. So the ones that get the little remnant pieces, those break down because they're not sufficient to maintain that cell. So we get one functional ovum versus 4 functional sperm. That makes sense? So big difference in the way the production of the egg and the sperm happen. One's just straightforward, the other one's very skewed, and kind of just gets one functional egg out. All right, so let's kind of look at this. Again, we've been looking at this in lab, so it never hurts to look at this stuff a couple of times. So here, of course, is your ovary, primordial. We know what that looks like. Primordial follicle. It's got our cell, the primary oocyte, and the little follicular cells are just like squamous epithelium. That's all it is. So just a single layer of squamous cells around the outside of them right here. Then once we are going to begin this process where they begin developing, you'll have several of these that will begin developing all at the same time. We turn into a primary follicle, and so for a primary follicle, the cells become cuboidal here, right here, and then you're going to end up becoming more multi-layered as well. You can see there's several all the way around the outside. So you've got the cuboidal right next to the oocyte, and then you've got the surrounding multiple layers that begin to develop. And so when we looked at those in lab, we saw them in a variety of ranges of development there. Then secondary, anytime you see the pockets, the little looks like air pockets, but when you start seeing the pockets form, that tells you you're at the next level. You're at the secondary level, and they would eventually go to where all the pockets are fused together into one big, large, single fluid, looks like a fluid-filled pocket called the antrum. And then, of course, you have your oocyte here. Now it's a secondary oocyte because it's gone through part of the meiosis here. And it's got the little surrounding granulosome cells just like it had before. Here, of course, we have rupturing of the follicle. So now here's all this part, all these cells stay, and here's the actual oocyte and the corona radiata, which is just the cells around that egg being released out during your ovulation. The remainder material is just called the corpus luteum, and then as it degenerates, it becomes the corpus albicans. So normal process as you go through of maturing. So in our lab, we're trying to identify those different stages: primordial, primary, secondary. The vesicular is also called the Graphian, and then corpus luteum and the corpus albicans. So that's the normal developmental process, but that doesn't really tell us anything about hormones. All right, so here with hormones, let's kind of look at this. So this is just for this process here. So hypothalamus, again, releases a hormone that targets the pituitary to release luteinizing and follicle stimulating. All right, so luteinizing, let's do follicle stimulating. Follicle stimulating hormone, well, I better not. Luteinizing first, comes over here, and luteinizing is going to be basically responsible for getting the production of estrogen going. So you can see here, it starts off as an androgen, which just means sex hormone, converts our sex hormone to estrogen, and so it basically begins that process there. I'm not really going to do the inhibit, okay? I'm going to skip that part there. Estrogen, and then you can kind of go here. Estrogen then helps to stimulate maturing here. Follicle stimulating hormone helps with the developmental processes that we've been looking at here, and our estrogen is going to help with the actual ovulation over here. This is not, the pictures look really similar like they did with the males, but it works better if you can look at a different graph to help see this, because you're going to have some surges of hormones to help stimulate our ovulation to occur. So let me kind of flip to this next picture, and maybe this will make a little bit more sense. I don't want that one. Here's the one I want. Y'all are going to go, good God. I don't want that one. So let's walk through this. All right, let me see which one I want to look at. Okay, so what you're seeing, because I have to sit there and go, what am I looking at? These are the two hormones coming from the pituitary, luteinizing and follicle stimulating. These are the hormones estrogen and progesterone that are going to be coming from the ovary, from this area. So we have there's basically 4 hormones we have to look at. So let's walk through just luteinizing and follicle stimulating. So it's got a cycle here. This is a 28-day cycle. So first few days, week, 5, 6 days, this is the menstrual phase here, right? So you can see we really don't have a whole lot happening here, so this is during menstruation. Then you're going to see here, right around 14 is considered the normal peak ovulation time period. You can see there's going to be a surge. This is called a surge of both of these hormones, luteinizing hormone and follicle stimulating hormone, and then those levels come back to kind of a normal level here. So kind of focus in on these two will surge to help stimulate ovulation. It's going to be your normal luteinizing follicle stimulating, all right? Now also what we have going on here, so again, menstrual phase here, in response to our levels of luteinizing hormone and follicle stimulating hormone, we're going to start getting an increase of estrogen. So this estrogen is a result of good old luteinizing hormone here. This is being released here. You can see again, estrogen peaks during ovulation. Now we have another hormone here that begins to rise after ovulation, and it comes all the way up and it peaks way up in here, which would be really to the point where are we going to have a fertilized egg and implantation, all this kind of stuff. And if not, then this is going to diminish off when we go back to the menstruation. This is progesterone. So where does progesterone come from? The corpus luteum. So corpus luteum does progesterone. So the estrogen is coming from our ovary, but it's being stimulated by luteinizing hormone. So let's repeat this one more time. So normal levels of luteinizing hormone and follicle stimulating hormone. The follicle stimulating hormone is stimulating basically the maturing of the follicles, and the luteinizing hormone is stimulating the release of our estrogen. About halfway through, they'll get a surge of both of those to stimulate ovulation. So here, then you see what is luteinizing hormone stimulating? It's stimulating the production of a hormone called estrogen, which peaks at ovulation, and then you can kind of see it varies a little bit. It's going to fluctuate some. And after ovulation, we have an increase of progesterone, which is basically setting stage for fertilization and implantation in the uterus. And if it happens, that's great. And if it doesn't, then it drops off again. So this comes from the corpus luteum after ovulation. And the corpus luteum would take over or keep doing the progesterone until the placenta begins. You have implantation and the placenta would begin doing that. So something has to carry that weight of a placenta until it's actually produced, right? Cool. I can tell y'all are enthused. That's okay. And this just kind of maps it all out. You can see with the progesterone, what you kind of focus on here is this is kind of the lining here. This is the uterine cycle. So this is the thickening of the uterine lining. So right here, it's shown kind of fuzzy because this is your administration where you're slopping it all off. And now we start building it, and most of this building that you can see here is a result of the progesterone that's peaking, is what's actually helping with all that. Okay, that's the last picture. You see, but that's just that separate one pulled out. I'm just looking to see if we forgot anything with this. They find that, just to kind of throw this out here, while I'm thinking of it, a lot of early miscarriages. Miscarriages that have, so miscarriages, 20% of pregnancies have miscarriages. 20%, which is a high percentage. So why do we have miscarriages? Couple of reasons. One of the reasons we have miscarriages has to do with this and the process. This process here has a lot of places where error can occur. Okay? Just a lot of processes, a lot of steps in this one where error can occur. But also what we see it's more common than you think is you have early miscarriages sometimes in that 8 to 12 week time period where they're so early. We know more about them now because we can detect pregnancies earlier than what we used to. Used to, you had to be further along before your pregnancy test would show up positive. Nowadays, they show up almost the same day as you get pregnant. I mean, they're exceedingly specific. And so a lot of people would have gotten pregnant and then been a week late and then had a heavy period and they actually had a miscarriage and they never knew they had a miscarriage. So because none of the conditions were prime, and so you had that problem. You also see the early ones that, again, 8 to 12, is due to low progesterone levels. So they actually go in nowadays, once they've targeted somebody who's had several miscarriages and they're all in that very early time period, they'll check their progesterone levels. And if their levels are lower than they would normally be, then they just supplement the progesterone during this particular time until the placenta is formed. And once the placenta is formed, they say, okay, you can quit that, and they'll take that away, and then everything's fine. So that helps with those early miscarriages. Questions? Anybody got questions? Y'all are so excited today. Okay, well then, I'm just going to get out of that, and we're going to talk a little bit over here about blood gases now. To go away. It's going to go all the way to 4. Come on, come on, come on. All right. Go back and just draw in the middle. Let's turn this off. one of the things that we like to try to talk to you a little bit about is acid and bases in the body and buffer systems and blood gases and those types of values. You can talk about it during all sorts. You just kind of have to pick when you kind of throw these out. But and there's a lot of variation with this. But I wanted to at least say, hey, we've looked at this stuff. We've talked about it a little bit. So everybody, and you kind of know pH, because we talked about this as well earlier this semester. pH, of course, goes from 0 to 14, right? 7 is considered neutral. We know that pH is a measurement of our hydrogen ion concentration, right? And as the hydrogen ion concentration increases, then our pH decreases on our scale and it becomes more acidic. All right? Conversely, as our hydrogen ion concentration here decreases, our pH will rise and we become more, and we'll use the term alkaline. You could say basic, but alkaline would be a little bit more appropriate term for us with what we're talking about. So one of the things that you see a lot in clinical settings is you have people come in and they'll say, okay, we need to get some blood gases to see what they are. So they're going to take these and they're going to do arterial blood gases, so not venous, we want arterial blood gases. And they're going to give us a representation of O2 levels, well, I'll put PO2 levels, carbon dioxide levels. They're going to give us a bicarbonate level, and they're going to give us a pH. So those are usually pretty much what you're going to get when you get a blood gas, those types of things. Some hospitals, the lab people do them. Other hospitals, you actually have people who specialize just in blood gases. That's all they do. And so those people come in and do these particular types of tests for you and stuff. If you have someone coming in and particularly have respiratory issues and you're trying to evaluate their O2 levels and what their other conditions are, these can be very informative, I guess you would say. So here's your normal range for your pH, 7.35 to 7.45. O2s are usually going to be somewhere around 98 to 100. PCO2s, your carbon dioxide, somewhere between 35 to 45, 45. And then your bicarb, somewhere between 22 and 26. And these are like millimeters of mercury as far as readings go. So that you're kind of like, okay, that's great, we got that. So what you have to do is when somebody comes in and you get these results, you kind of have to figure out what are they. Well, the very first thing you always do, look at your pH. Look at pH. Because that's going to tell you the first thing. So let's say we're going to go through 4 examples. We're going to go through, is it acidic, and 2 examples of that, and then we're going to go and look at, is it alkaline, and 2 examples with that. All right, so, and I always have to do this because I never keep this memorized, and I do apologize about it. Let's do acidosis first. So, pH less than 7.35 equals acidosis. They have an acidic pH. Acidic pH. Next thing you're going to do, second, you want to look at your carbon dioxide. Look at the carbon dioxide level. So if our carbon dioxide level, if PCO2 is greater than 45, it's telling me my carbon dioxide level is higher than normal in the body, then my problem's with my respiratory system. This would be somebody coming in with pneumonia. Classic. So we're going to put over here pneumonia. Really good classic example there. So that one's pretty good. If though my PCO2 is normal, then I have an acidosis that is not due to the respiratory system, right? And so what that means, I have some sort of metabolic acidosis. Metabolic acidosis, where I'm looking for something. Do I get another thing? Good, I don't. Okay. Now with that one, I usually will see my bicarb, I always keep forgetting what my H is, less than 22. Bicarb is going to be less than 22 with this. Okay? Because what it's telling me when I see this, it means my pH is low, my carbon dioxide is not a problem, so my bicarb is my buffer system. Remember bicarbonate, the carbonic acid bicarbonate buffer system? So the idea behind this is if my bicarbonate, if I have a whole lot of hydrogen ions, it's gonna combine with that. So now what this is telling me is I've done that already, it's still got a low pH, and my levels of bicarb are beginning to be low. So this is a metabolic acidosis. Metabolic acidosis. So common conditions that cause metabolic acidosis would be something that produces a lot of metabolic acids, like diabetes. So ketone acidosis is metabolic acidosis. So someone who's diabetic would be indicative of this person over here. And also another one that's super, super common, and I apologize, I know what I want to say, and then I go, ooh, is that right? No, yeah, okay. All right, so we have that one, because this is excess ketones, is what we're seeing here. And do y'all know what ketones are? Yes, no? So ketones are from the body metabolizing things like protein when it doesn't have sugar available. The cells can't get to the sugar because there's no insulin to tell the cells the sugar's there. So they have to look at a different energy source, and so they're utilizing proteins or whatever, and so this is a metabolic acid that is a buildup of that. So it would show up in your analysis as a positive as well. You can get positive ketones and everything else with that. All right, so that's one way you get, that's one condition. You could also have, like, what about just plain old excessive, I don't want to say excessive, excess exercise causes an increase in lactic acid. Now remember, lactic acid came from anaerobic, it's been a long time, anaerobic metabolism. You're really working those muscles so hard and so strenuously that they're not burning, that there's not oxygen there, so they're not burning glucose. Excuse me, I'm saying this all backwards. There's not oxygen there, so instead of burning through, fats and some of your other things, they're burning pure through glucose, and because there's no O2, the pathways change, so you get this buildup of lactic acid as that byproduct of that metabolism. And it accumulates in the muscles, and that's what contributes to muscle fatigue and the muscle soreness. But it also is an acid that's still in the body that's got to be flushed out. We've got to get it out of the body. So while it's in the body, it can cause a metabolic acidosis. So we've got diabetes, ketoacidosis, and we have exercise-induced metabolic acidosis due to lactic acid accumulation. You can also have another one that's very common. Acetic acid is another type of metabolic acid, and it comes from alcohol. So someone who's really been on a serious, an alcoholic, someone who's chronically drinking an awful lot, they can come in and present with a metabolic acidosis, but it's due to the accumulation of acetic acid from the breakdown of alcohol metabolism. So 3 that you would see, the most common, this one, is alcohol. That's our most common one. Second would probably be your diabetes. If you found somebody who's starving, someone starving, they would also be in this category. You could have acidosis that over here too, malnutrition, like hunger strike kind of thing, because their body's metabolizing itself, and it would be, you would get this part, you get ketone bodies being produced. All right, so those are acidosis. So we need to look at the opposite then, alkalosis. Okay, so 2 conditions here. So pH would be greater than 7.45, and we say they're in alkalosis. So we want to define 2 types, respiratory and metabolic. So we have metabolic and respiratory here, we have metabolic and respiratory with alkalosis as well. So let's do, what do I have listed first? Respiratory first. So with respiratory alkalosis, you're going to have actually low PO2s. So this one, your carbon dioxide level is going to be less than, what is it, 35? 35. Less than 35. This is hyperventilating. Someone who's just doing the pant machine going. It's not very common, but you do see it occasionally. We would say in most cases, respiratory alkalosis is not associated with a disease process. It's a condition, but it's not really an underlying disease like we have with some of these other ones. Now, metabolic means that then, same kind of thing, the CO2 level would be okay, but our bicarb would be greater than, what is it, 26. And you're going to see this one, again, the alkalosis, neither one of these are super common, but you can see it from people who take a lot of bicarb, a lot of antacids. So we could say excess of that can cause this condition, excess vomiting and diarrhea. Tend to also cause it as well. Oh, and the last one is constipation. So again, you don't see this one nearly as, well, neither one of these are really terrible, but this one, you could certainly, I mean, you've got disease processes actually happening here. And you have some people who eat Tums and bicarbs and antacids like it's candy all day long and stuff, you know, just continual, and they can certainly put the body into an alkalosis state and stuff. And of course here you're losing a lot of electrolytes here, so that's a lot of the problem here. So you'd be losing a lot of some of your acids that way. Okay? Now, this is kind of the most basic breakdown when you look at blood gases. You got your pH, is it normal, is it low, is it high? Then you look at the carbon dioxide levels because you're trying to figure out, is it a problem with the respiratory system? Then if it's not a problem with the respiratory system, then you know you're looking at metabolic problems. Now, what you'll end up seeing when you actually are in clinicals is that some people will present this way, but you'll also see some crazy variations depending upon what disease they have. And there's a term called compensating. The body will go into one of these conditions and then try to compensate for it. And so then your values get screwed up. Your values can look almost normal and you're going, what is going on with my person? I am so confused. And it's because they've been thrown into this, like somebody who has maybe respiratory acidosis, so their CO2 levels are really high over here, might try to compensate in some way, like by hyperventilating to try to blow it off, and so then you might hyperventilate enough that they get one of these numbers back down, but they're not, but you know they're not right because you can see they're hyperventilating. I mean, so you're gonna have some that you're gonna have to go, okay, here's my rules, but what are they doing? Because one part of the body will try to compensate for the other side to try to get those numbers. The whole goal, they don't care so much about these numbers as you're trying to get that pH range back up to where it needs to be. So you have to kind of be mindful that it's not always going to follow this exact rule or present exactly like this just because of that. All right. I think, believe it or not, that is it. I mean, we can get in and talk about water balance and talk about some other stuff, but I'm like, can we not be done? So that's it. That's all our lectures. So we just have, on Wednesday would be a review day only, and then we test that next week. So we've made it. We've made it to no more lectures. That's awesome. And let's see, case studies. If you, I think.